Practical skills for AS level. In an experiment investigating the effect of one variable on another, the independent variable is the one that you change and the dependent variable is the one that you measure. All other variables should be standardized, kept constant. The range of the independent variable is the spread from lowest to highest value. The interval is the distance between each value in the range. Temperature can be kept constant or varied using a water bath. pH can be kept constant or varied using buffer solutions. The accuracy of a measurement is how true it is. For example, an accurate measuring cylinder reads exactly 50 cubic centimeters when it contains 50 cubic centimeters of liquid. The precision of a measuring instrument is how consistent it is in giving exactly the same reading for the same value. You can increase confidence in your results by doing replicates and calculating a mean. Results tables should be constructed with the independent variable in the first column and the readings for the dependent variables in the columns. Units go in the headings, not in the body of the table. Each value should be recorded to the same number of decimal places. This is also the case for any calculated values. In a line graph, the independent variable goes on the x-axis and the dependent variable on the y-axis. Headings must include units. Scales must go up in even and sensible steps. Points should be plotted as small crosses or as encircled dots. Lines should be best fit or ruled between successive points. Do not extrapolate. Bar charts are drawn when there is a discontinuous variable on the x-axis. Bars in a bar chart do not touch. Frequency diagrams or histograms are drawn when there is a continuous variable on the x-axis. Bars touch. Conclusions should be short and to the point. They should use the results to answer the question posed by the investigation. They should not go beyond what is shown by the results. Do not confuse conclusion with discussion. When describing data displayed on a graph, begin by stating the general trend and then describe any points at which the gradient of the curve changes. Quote figures from the x-axis and y-axis coordinates for these points. Do not use language suggesting time, e.g. faster, if time is not shown on the x-axis or y-axis. Show every small step whenever you are asked to do a calculation. Do not confuse mistakes with experimental errors. Mistakes should not happen. Experimental errors are often unavoidable, unless you have the opportunity to use a better technique or better apparatus. Systematic errors are those measuring instruments. Random errors are those which vary in magnitude and direction during the experiment, and may be caused by difficulty in standardizing variables or in making judgments. When asked to suggest improvements in an experiment, concentrate on the main sources of error and suggest ways of reducing them. When making drawings from a microscope, a low-power plan should show only the outlines of tissues and no individual cells. Be prepared to go up to high power to get more information about where one tissue ends and another begins. High-power drawings should show as much details as possible, including details of individual cells. Lecture notes for